So I'm full professor at the Department of Chemical Engineering and senior researcher in the Instituto de Ciencias y Materiales de Aragón. I'm glad to share this afternoon with all of you, people who is attending today in this room, and those of you that are also connected online. As you know, it's Thursday, on this the last Thursday of every month. La Cátedra Sanca de Nanotecnología grants a lecture given by a spin-off company. So that's the reason why today we have a lecture. And the speaker for this lecture is Samuel Sanchez. It's difficult just to present Samuel because he has a brilliant CV. So I will try to just to summarize just a little about his work and his hours and so. So Samuel obtained the PhD in 2008 at the Autonomous University of Barcelona. Currently, he is an ICREA professor in the Institute of Bioengineering of Catalonia. And previously, he was in the Max Planck Institute in Stuttgart. He has plenty of hours. Maybe the most interesting to me are the MIT Top Innovator Under 35. He has several Guinness World Records. I'm curious about those hours. Also, he was awarded with the Princess of Girona Scientific Award in 2015, the Banco Sabadell Foundation Award. He was also elected member of the Joint Academy of Spain Science 2020. About projects, he was awarded with several prestigious projects, 2 ERC and a starting grant in 2013 and a consolidator grant in 2019. So as you can see, he's a very brilliant research. He has published more than 165 papers, and he has an H index of 72 with 8 patents. So besides of all of that, he's the co-founder of this NanoSpin-off, NanoBots Therapeutics. And that is the main reason why he's today with us, just to show what is about this spin-off company. So Samuel, thank you very much for staying with us today. I just give you the floor, and you can have like 45 minutes just to share with us the knowledge of the company, and then we'll get a section of questions for you. Thank you.
Yeah, thank you so much. Thank you, Victor and Marta, for inviting me here today. It's my pleasure to present the activities that we do the Institute of Bioengineering of Catalonia, but also the activities with regards to the NanoBots Therapeutics. I'll be sharing my screen. Let's see. Right. Okay, I did something different now. Right. Is it okay now? Do you see well? Because I see twice.
Nice, perfect. Yeah.
Yeah. Okay. Good. So yeah, as I said, I will be presenting the work that we do regarding nanobots. And NanoBots Therapeutics is the spin-off. NanoBots is the technology, or nanorobotics or nanomodel is basically what you see here, and this this type of particle that are decorated with enzymes. And these are therapeutics in motion, and this kind of our law or leitmotif, right? Like what you are going to see today is a lot of things moving around, and what we want to do is to have a smart, efficient, and very active drug delivery system that will be continuously in motion. So I'm a career professor here in Barcelona, which belongs to the Institute of Bioengineering, is a part of BIST. But also, like the main reason, I guess, because of this this Thursday's Nano spin-off is because in 2013, I was one of the co-founders of the spin-off NanoBots Therapeutics. You can see the website here. You can follow us. And since January, actually was last month, I also have to jump from CSO and together with the CSO, I have also the working load and the task of being CEO and interim CEO of the company. If you want, we can. I want to leave it open for questions later. If you agree with regarding the structure of the spin-off, how we found it, and if anyone has any curiosity regarding that, I would be glad to answer the questions. But today, I think the idea was more regarding the science behind NanoBots, what we do together with IBEC in the company, how we plan to bring this technology forward to the clinic. So one of the motivations always we like to start with this movie or the book from Isaac Asimov, This Fantastic Voyage, in order to where they wrote and they presented a troupe of doctors going inside a living system, like a human being, right? Like trying to heal and to cure this patient from the US Army. And having an efficient delivery system has been important for many years. And one of the ideas was claimed 60 years ago already, is to do something which is right now becoming a reality, which is the precision medicine in nanomedicine. So what we want to do is, in fact, having a system that moves actively. And if this will represent a tumor, and that was presented almost 10 years ago in El País, but from one where there was an interview where I was talking about this idea, right? Like, we don't want to go only to the outside of the tumor. We want to drill and we want to penetrate and we want to go to the center or going deep inside. And in fact, at the end of my talk, you will be seeing how almost 10 years later we managed to do that. So this is very important, right, in precision medicine nowadays. But still, there are many challenges on nanomedicine for crossing. Actually, one of them is because we need to cross a lot of biological barriers, and that was very nicely summarized in this review from Wilson Poon, where they said, okay, once you get here, you inject. Mainly, they was talking about intravenous administration, but anyways, you can find protein corona formation that will stop all the moieties of the functionalities around the particle. And this is not good. You can have sequestration. You can also get to the transport target cells. You can see there's a lot of extracellular matrix. There's a viscous environment that will stop nanoparticles to go through, right? You can find also blood vessel extravasation that you need to tackle. And also, once you get to the cell, right, you need to fight against, work on this endosomal escape and how you go to the organelle targeting. So there are many challenges that need to be addressed. We try to do some of them, right? We had a study just reported in nanoscale about protein corona formation, how the nanobots reduce protein corona and still keep moving. Also, we just published in small how we can move in very viscous mediums, synovial fluid. And actually, we have another paper in preparation, another subtopic on how we should understand the endosomal escape. So what is happening here, right? When you make a nanoparticle, when you make a drug, and this drug needs to get into the cell, they might encounter all these biological barriers that with the stock people have done, and you know for sure, right? In this course of nanotechnology, right, and nanomedicine, that you make nanoparticles, you encapsulate the drug, and you can cross. But probably once you get there, your nanoparticles with the drug will get trapped into this mucus layer. So what we want to do is something actively that moves, gets to the target side, but also can interact with the extracellular matrix on this mucus, right? And this will be another biological barrier. This will modify the rheology, the viscosity, and the composition of this mucus. And this is something that we didn't invent it now. Like, nature has done it for millions of years. So Helicobacter pylori can cross the mucus layer of our stomach thanks to not only the motion, but a chemical reaction which is on the outside of it. So the bacteria has the urease enzyme that you will hear a lot about this today. The urease enzyme takes the urea, increases the pH of the environment, and then reduces the viscosity, and then can move forward into mucin gels. And this is a very important concept that we are taking up in our technology. So how do the nanobots look like? Which are the components that we need in order to make a nano system that will move? We need two components at least. You need a chassis and you need an engine. The engine will propel the particles forward, and a chassis will be, again, a nanoparticle, right? So again, we will be looking at the nature today a lot. And if we look at the cancer cells, bacteria, viruses, not only the size, the shape, but also like the structure on the outside. We can copy it. We can make micro-sized particles. We can make nanotubes or nanorod-like bacteria structure. We can make very small particles like virus-like structure. But one of the main ideas, and that's something that we're working in the company, is not to use, because at the research lab, we were working on 8 up to 10 different type of particles that you can see in our publication record, right? But basically in the company, what we're doing is taking a particle chassis that will be FDA or EMA approved. So we want to de-risk the path to the clinic, right? So then at the regulatory level, this will be much easier to scale up for the CMC and also to be accepted by the next regulatory agencies. So this is one of the main features here, having a very well approved and standardized particle. So the second component will be the engine. How to make this particle move? How do we give this particle energy in order to move in this very complex fluid? So there are many different ways that people have used in our community, but we are working on chemical reactions based on catalytic reactions. I will show you a couple of examples how initially we started with platinum, but now we're ending up with enzymes. So why is that? This is because we want to copy nature, right? So bacteria not only use the enzymes on the outside to change the environment, but also use catalytic reaction in order to move, to convert the chemistry which is around, right, into motion. So that would be an ideal system. You have your chassis, you have your engine, you put into a solution which is chemically rich, right? So you have the fuel in your body, and this is already autonomous enough, and then they can move. Okay, so in our field, people have started years ago. In maybe this is actually now 20 years since Maluk and Sen started making this rod-like structure using platinum. You can see two different materials. People call it like Janus particles, like two phases of God of two phases, where you have the catalytic material here. They will make a chemical reaction and move. You can make spheres, plates, rods. You can make tubes. You can make stomatocytes or polymersomes. You can make Janus particles at the nanoscale and so on. But always there was an asymmetry, and always in this case, historically it was used based on platinum. So this is what we were doing 10 years ago, having a large tube of about 20 to 5 microns, 5 to 10 in length, 5 to 10 microns in diameter, where the platinum is inside, makes in peroxide, makes a chemical reaction, move the particle forward. But the problem here, and this could be used for different applications like mixing fluids, sensing, and also environmental applications. That was part of my starting run. But then there was a limitation or when you want to move in vivo, right, on biomedical applications. So this is making bubbles. You don't want bubbles in your body. This is peroxide. Peroxide is toxic for the cells mainly, and you don't find in a large concentration in your body. So we wanted to change to different systems. So we started to miniaturize, to reduce the size of these nanomotors. As I told you before, we wanted to mimic this asymmetry where having the two phases. This was complex from the engineering point of view. So to say the efficiency was not very high. We don't make bubbles, not so impressive. But you could start to think about collective motion here in a way, how particles interact with each other and move in fluid. So you can also miniaturize from microns down to a nanoscale. So that was one advantage. You can have something really small on the nanoscale or hundreds of nanometers, and that's already going towards nanomedicine. Now, we needed to tackle the problem of the platinum and peroxide combination, right? So in order to overcome this problem, like we change, or problem like we provided something new, which is using enzymes as an engine. Enzymes convert endogenous fuels. You can have thousands of them in your body and in cells. So this is a very efficient system to get motion. And so when I was already, at that time, I was finished my PhD, going to Japan for my first postdoc. So I saw this paper from Ben Feringa, who later on got the Nobel Prize, right, in 2016, nanomachines. So he was taking a multi-wall carbon nanotube, as they claim that this is somewhere here, maybe these things moving, making bubbles. There was combining two enzymes, glucose oxidase and catalase. So you take glucose in your system, convert it into peroxide locally, and then you generate the oxygen that you will see into that. This was not a very efficient in terms of motion, and you see there's a lot of kind of garbage somewhere around here, and you can have to put oxygen to generate oxygen. So it's a bit tricky system, but motivated me to take the long tube that we were doing in the beginning, converting, changing platinum for instead of using that to change by the catalase enzyme, just only one enzyme, right? So with this one, we could convert peroxide into oxygen, making oxygen bubbles, but a much lower concentration than before. So that gave us some hint that this could be a useful application. So now let's combine the two of them. Well, these are some examples that people have used it, also enzymes, not only us, to have long-range propagation of signaling and copying the Helicobacter pylori and making chemotaxis. So I will jump into that, combining the two things that I told you before. Right? Now, instead of tubes, we take particles in the micro nanoscale, but this should be biocompatible. Now we take the second component, which is the enzyme. What enzyme are we dealing with today in this talk is urease. So urease takes up the urea into CO2 and ammonia. Instead of the catalase is over already, so we'll move to this one now. And instead of making something physically, asymmetrically different, right? With the two phases, we make something simple. We make a very, let's say, bad functionalization. So we take glutaraldehyde, we cross-link the enzymes, and we actually, by super resolution, could observe that if you label the enzyme, every dot here is a single enzyme, right? So you can see, this is from the top, that there are some asymmetry, some gaps around the particle. So in a way, it's like this, right? And this asymmetry can be modeled by some computational codes on the density map, and you can see the blue is like high density patches. So every single particle that we produce has a different asymmetry, totally stochastic. We cannot control it, but the truth is that all the particles do move, and they move like this. So you have the control where there's no particle moving. And here, when there's urea in the media, you have urease enzyme. So this is moving, as you see here very clearly, totally autonomous. Okay. So what is next? And you can do this for any type of particle, as I show you in the initial slide, right? You can take nanoparticles, you can take microparticles, and you can take also these nanotubes. Okay, so by the way, Victor, this is actually the one in the Guinness record of the smallest jet engine in the world. So we have, this is 220 nanometers in diameter. I don't know if you see, I mean, we have the award behind me here. Anyway, so we have totally biocompatible motor right now. So what can we do with them? So the next thing is to add, so we do have two components I told you so far. If we add a third component, therapeutic one, or targeting, or for different applications, like I export here, like transport, sense, act, or target. And this is actually within our patent, which has been accepted. Now it's in 9 different regions, and it's accepted clear patent in Europe. So the first thing to do is to demonstrate whether active transport is better than passive transport, and we did that. Towards cancer cells like HeLa cells, we showed an enhanced delivery system. Then we also functionalized them with DNA nanoswitches, this together with Francesco Abizi's lab in Rome. So the nanobots were moving and sensing the pH and sending a different color depending on the pH. That was very, very nice. And if you want to move from sustained release to triggered release, this was done together with Ramon Martin in Valencia. So we have the nanomotors coated with molecular gates that will open only a lower pH than well, I was 5.5. So when there's inside cancer cell, and the last one coming towards our application. So we target, we coated the nanoparticles with the nanobots with targeting antibodies for cancer spheroids. And then you see here already the key point of what actually the spin-off is working on bladder cancer. So why bladder cancer? I told you in the beginning that you will hear a lot about urease, right? Urease takes up the urea fuel, which is in the solution. But in which solution we have the fuel? In the urine. The urine is in our body. So from the technology point of view of nanotechnology, this was very appealing, right? We have a system that moves inside our urine, in urine solution, inside our bladder. But then we started talking to clinicians, and then they referred that cancer, bladder cancer, is a clear medical need, because it's the 7-month cancer, most common cancer. But not only that, I think the important here is that is the one has the highest recurrency rate. So if you happen to have bladder cancer, you might go to hospital like almost a dozen of times, because diagnosis and the treatment are right now not so efficient. So the tumor will grow. And that makes it the most expensive cancer to treat. You will need to go to hospital over and over and following this very tedious and consuming treatment. So we started 5 years ago working on in vitro and in vivo experiments. So we demonstrated the enhanced delivery. As I told you, like we have here, we see this actually is 2D cells. You see the nanobots inside the cell. We saw an enhanced delivery in 2D, but we also saw the penetration and targeting in bladder spheroids. How we did that? So you look at the bladder. Bladder is a 3D, bladder tumor is a 3D structure, that kind of a spongy-like that is growing there from the urothelium. So what we did is to take the silica nanoparticles, we coated with the urease, but also with the anti-FGFR3, which is normally overexpressed in these type of cells. And then this spheroid we can target and break it and stop tumor progression. So these are some of the results. I just summarized the whole paper in one slide, where you have the spheroid here, hundreds of cells, bladder cancer, and then if there's no urea in the medium, you're not going to see any red signal, fluorescent signal from the nanobots inside, just very minor one. But if there's a little bit of fuel of urea actually here, nanobots will move. They will encounter, like they will reach to the tumor, and they also will penetrate, right? So this is what we see here in clearly enhanced, where the nanomotor contain urea and also the antibody, compared with the case where there's no antibody. So we've seen 14 times increase in penetration, but we have seen also a reduction of the tumor progression, which is extremely good. Okay, so this motivated us to move to in vivo. And the next question in the state of the art is like, can we actually see them in a living system? And that can be done by intravenous, but also the most interesting part is intravesical administration, because this is what they do in the clinic. And this is what I will tell you today, right? But in order to get there, single particle do nothing. So we want to again copy nature. So one of the ideas is to see if we have a high density of nanobots moving collectively. This is not, of course, our nanobots, right? These are birds in the sky moving together. So the only motivation of this slide is to convince you that what we are doing is the following, which in a way resembles the motion of flock of birds, right, or fishes, right, or swarm of fishes moving at the macro scale, but doing this at the nanoscale. So this is what we plan to mimic. We still need to understand why is this motion and this type of phenomena, but has been very interesting in all the ways, like because you can see a clear difference when there's no urea. So what we are doing here is we add a drop of these nanoparticles in a petri dish, they will just expand. But if there's urea in the middle, they start this vigorous reaction moving in 3 dimensions that is happening all the time. So if we want to see them, we need to label them, right? And we are going to use PET-CT imaging. This is the Silvio Magún in the Basque Country. So we had 2 different strategies: the fluorine directly to the enzyme, or iodine physically absorbed on the gold. And we use microfluidic systems to first track in vitro the motion. So we have seen the 2 cases here. The right side, you will see when there's urea, on the left side when there's no urea. And you can see some difference. The more we challenge the path, the higher the differences will be. As you can see here, this is the swarm of nanobot imaged by PET-CT, by PET, right? And you can see difference in the migration. So you can quantify that. That's not a big story, but now let's go to what they do in the clinic, right? Is what we are gonna do here with the animals. So we're gonna empty the bladder, we're gonna administer locally, like intravesically, drop here, and then there would be two-phase formation between the nanobots and the liquid. But if the nanobots are moving, they will be mixing. If they are not moving, they will just sediment, as it actually happens in the clinic when they ask you to lay down, they administer here in the bladder, and then the liquid will sediment, only stay one side. They will ask you to rotate. So here, the red cloud that you are seeing is the cloud of nanobots. So you can see here when the nanobot moving in urea in the bladder, they reach the whole volume. This is a CTA reconstruction for the winning case of the nanobots, and the other 3 cases are the control. You can see that over 45 minutes of experiment, this will stay only on the one side. So this is not efficient. So actually, you have something that moves inside that reach the whole volume. This will enhance the probability of the drug to get to the tumor, but there is no tumor here, right? So, well, this is images on what you can see and what you can do with the PET-CT, the image, okay? So the next question was, can we treat bladder cancer tumors in vivo, right? No. After that publication, now we got the neural model, we generated it, and then the next step was to see the tumor grow in the monitoring, administer the same day after 14, 15 days, and see whether they accumulate in the right place and whether we can treat it. So this is part of our consortium from the Kasha Research, Kasha Health, between clinic, UAB, Fibio Maune, and us, but in the last step, IRB, you will see very fancy imaging techniques that they supported this project. And this was published very recently, so I think this talk is also very timely because it was about a month ago when we published this in Nature Nanotech, where we are using radionuclide therapy to treat bladder cancer. And this was appeared in a lot of media recently. So these are multifunctional nanobots because we can use them for imaging with fluorine. We can use it for therapy using iodine. And this, again, the cloud of the nanobots moving collectively and reaching a large area and large volume. So the first thing was, again, to generate the tumor. This is not a big story here. The histology to show that the bladder is not damaged by the presence of the nanobot. The next thing was to administer the nanobots here. This is kind of crowded, but you will see in the upper layer, upper row, you will see the MRI, where we have the control. There's no tumor. Then when we have BSA nanoparticle, we mean when instead of urease, we use a different protein that doesn't provide the motion. So they should not move, they will sediment and not accumulate. As you can see, PET imaging, there's slight imaging, but there's no significant difference between the control and these two. And then when we go to the nanobot, which is moving in urea, already is active, there we administer, and actually you can see, right, there's a huge increase in the tumor accumulation, and you can quantify with very significant difference compared with the others. So first story I told you, like nanobots can move in the bladder. Second, they reach to the tumor and they accumulate there. Now the question was, where do they stay? Do they accumulate in the outside? Do they penetrate? For that, we did ex vivo. We took the mice, the bladder from mice, and we brought it here to Barcelona to IRB. So they have this nice imaging, this light microscopy, which is crossing. Yeah, it's going, cutting just by light, not damaging the sample, like up and down. And you can distinguish between the mass, like on the tissues. You can see in gray, and you can see the scatter signal from the nanoparticle, from the nanobot. So we can almost identify a single particle level, almost, right? But in any case, you can see all this yellow, red signal, which is specifically accumulated, and can see here how deep it gets because we see the tumor, and this is the healthy tissue on the right side, very smooth. So if 95% of the nanobots do accumulate at the tumor and they penetrate. So this was like, for me, fascinating results. You can also see different type of imaging, and you can quantify these blue colors is tell you the signal where there's no particle, red and yellow. It would particles identified. Actually, we can go deep into the whole volume of the tumor. Okay, so this is now we move to the therapies. We said now that we get there, let's treat the tumor. So this is just to show that anyway with the radionuclide, the animals don't lose weight, so they're healthy. And now these are the experiments. We validate the efficacy of our treatment. I told you that if you happen to have bladder cancer, you will go like 10, 12, 15, 14 times to the hospital. But here, only with one intravesical administration, we managed to stop the tumor progression, first of all, right, by changing now the imaging radion or like isotope for the radionuclide, isotope iodine-131. So in here, low doses, we have a stop of proliferation, but if we increase the doses, we can reduce down to 90% the tumor progression. Okay, so this is again great because it's one administration, and second is like because still we call high doses, this 5 times lower than the one used for steroids and radiotherapy in humans. So this is pretty low. Okay, so these are different way of seeing the data, how we, by different type of experiments, we can reduce the tumor volume compared to non-treated, and how we can flip the progression. So if you don't treat the tumor, it will keep growing. And if you treat with nanobots without any therapy, it happens like this, then you can stop it and then you can reduce it significantly. Okay, so far I told you about bladder cancer. We have, I think here is very important as well, talking about the spin-off, is if you want to bring something to the clinic, this always happens like this, but in the spin-off much more, you need to talk to the clinician, you need to have their advice. And in this case, not only that, we do have a collaboration. We have medical doctors in our lab. We change samples, we get samples from the patient. Actually, this belongs to patient-derived spheroids or organoids that we do have in the lab. So this collaboration with clinicians and the experts are extremely important. So another project that we're moving forward, because nanobot therapeutic and the lab as well, like we want to expand the indications, and we are actually working in a platform technology, right? So this platform should cover different indications like bladder, mucus crossing. So in this case, we have another student, Medice, who is working in a ERC proof of concept with Brown Hospital for another indication. I told you about the mucus, so we can cross mucus here with a different enzyme and different type of particles, where we can generate the bubbles and cross it. We also have one senior scientist in my group is working together with Ghent University and the pharmacy department in ocular diseases. So this is very important because, again, you see it's a very complex matrix. It's a very viscous media where drugs need to diffuse and expand and reach actually to the back of the eye. And this is not straightforward, and we are having very positive results in the direction. Also, Noelia published this paper, and we have together with Fundación García Cugat here in Hospital Quirón in Barcelona, project on how to disperse growth factors inside the knee. And in this case, we published macromolecules as a model example in synovial fluid. We are doing this in vivo as well at the moment. And we have another application like bone healing, skin, infection in the skin when you have wound there and biofilm. So we published this together with UPenn and the MIT groups from a couple of years ago. Okay, so this to show that there's a clear platform technology. There's not only one indication, and that's what investors want to hear from us when we go to the rounds and to raise money. So here's our contribution in the group, or from our group to micro nano robots. So in 2010, I started with this story I told you about before. So taking a long tube, putting an enzyme inside. So that was one of the pioneers' work on enzyme micromotors. We use this to drill into cells, but that was still toxic for them. So it's just a model example that we can drill into that. Then other indications, we also patented this technology. We had the ERC proof of concept on that, how we use the bubble propulsion, this self-mixing that we are seeing now to clean water. Also, you might have seen like SpermBots that was continued with Mariana Medina, Veronica Maddames in Dresden with Oliver Smith. So, but back 10 years ago, we trap and like sperms, and we use this for propulsion. But this was another project that they are continuing there. And then we moved from transition from large tubes to more nanoscale. So this was one of the first papers on nanomotors with enzymes, the fundamental work on patchy particles moving into spheroids. And very recently, already in this decade, we started working on, as I told you, different type of particle, MOF, lipobots, nanobots in vivo. We do also 3D, breaking fibers, viscous media, and now the work that I presented today, also how we are one of the first ones treating cancer using nanobots in vivo. So we are proud of this evolution and this transition here to more realistics, and then it made me eager to jump into this adventure of why not to create a spin-off. There are many reasons. Like one of them is to ensure that what you have done in the lab can be like reaching to the clinicians, to the clinic, reaching to the patients and to the market. To protect the IP, this is very important, and sometimes one of the few ways to do that is to find private investors who will keep protecting your patent, because in Spain it's very difficult to pay constantly. I mean, this is very expensive, so we have an amazing patent, I think, which has a lot of potential, so that's why we created this spin-off. But you cannot do this alone. This is very clear. This is the team by 2024. So as I said, this is the management, like Marta, she's the COO since January. She has experience in BioCat, IBEC Tech Transfer, and last year's in Ferren, the pharma company. And this is my background, you know, well, from the introduction. So since a couple of weeks ago, we know that Marta is our first PhD industrial, and then also Daniel applied for the top scale. So hopefully he can also be involved in the company, but he's already involved in some of the activities. But then when you build up your spin-off, you need to be surrounded by experts in completely different fields, not only the science, because the science is done here. I don't have the pictures of everyone involved from my group. There's a group of 5 to 6 people involved in different sub-projects that are providing with data and providing also with a clear collaboration with NanoBots. But you need experts in pharma, drug development, finance, strategy, and we do have them. So Andres has been working for more than 25 years in Almirai, Ferrer, and different pharma companies. He brought 7 drugs and their products to the market. Isaac has worked in PAC or Aluz, and he's CFO of different companies in his background. And Daniel Oliver is the director of Capital Cell and also founder of NARA Venture Capital. So we have also gathered a team of oncology experts and international advisors. We have also experts in CMC, enzyme production, person from EMA in nanomedicine, and of course, Jordi and Esther have worked a lot with us in radionuclides in vivo experiments. So we have also the funding here. So what is our strategy? And now just to conclude, investment opportunity, where are we now? So we had Rosen in a thing, people said here from the statistics that in the worst moment in probably in history, because we of the biotech investment was in 2023. In 4 months, we rose after building the company, €500,000 of proceeds, but that's not enough because now we need to move to the next round series, like will be a seed or pre-series A. And of course, you need to apply for a lot of non-dilutive funding, and we are waiting for some of these solutions. So what we want to do is to bring this MBT 101, this first product for the first indication. Now we are in the lead selection, and we want to bring into the candidate, IND, and also the clinical trial. So we expect to get to the first clinical trial in 3-4 years. But also the next round of investment will help us pushing the second indication, or the second product of the first indication, and the first product of the second indication moving forward. Because again, this is a platform technology that needs to be developed. So with that, I think I'm finishing now, but I could be explaining a lot of the things that we are working on and the scope of the company, but is clearly in the line of the research that we have done over the last years, thanks to all these people at IBEC. So there have been two main groups, people who started, you see their names here, started on the enzyme nanoparticles, and then all the collaborators and people who are nowadays, and even more people, because it's changing very quickly, pushing forward this technology. So I think with that, I thank also the funding agencies, and I thank a lot to you for your attention. If you want to contact us, here's our website. I think that's all from my side.
Okay. Okay, thank you, Samuel. It was an amazing lecture. Yeah, I think we can have a very good inspiration for our research. Just considering your success in this kind of research. So now it's time for questions. We have several questions here in the room. So maybe you can.
Can you hear me?
Samuel, can you hear the?
Okay, thank you very much. Very nice talk. So I have a very practical question regarding the nanobots from a regulatory point of view. Are they medical devices or drugs?
Yeah, that's a very timely question. Actually, we'll hear from that tomorrow 9:30 because we're waiting for our regulatory pathway. This and clear, I will tell you that this is not based on our understanding. This is not a medical device. It should be, but also it's not a new drug, right? So the point is here is that it will be more like so we do have a vehicle, right? I'm not sure, and that is as I said, that's why you need to contact regulatory agency, right? Like consultant agency, so we can give you advice on this. So it depends on.
Yeah.
I would say I would love to go into the easiest definition, no? So
Yeah.
that is clear, right? The point here is that we do have three components. And to my surprise, like as a scientist, I would say, well, three are not too many, but from the regulatory point of view, three are a lot. It seems so. We do have the particle, we do have the enzyme, and then we have the drug. So this might be a device. These but it's not a drug per se, right? Because we probably could use a drug which is already accepted, right, through regulatory, even if it can be in the market. So yeah, it's a very good question which I could not answer. Maybe tomorrow at 11 I can answer to you after my meeting. So
I hope they give you the answer you want to hear, to be honest. And if I may, another scientific question. So when you monitor this reduction in the tumor volume of the animals that you treat with the nanobots, for how long do you monitor? And you see that the tumor is decreasing and you don't see any relapse in time?
Yeah, so the experiment was done, I would say, in 1 hour. So the treatment was staying there, the nanobots stay for 1 hour, then they empty the bladder and then the image afterward. So you know you need to empty the bladder from the urine, and then whatever we observe is what it remained there at the tumor site. What we have not, I don't know the second part of your question, if you said about the relapse or
if you monitor the animals for like weeks or so,
exactly.
That is not growing back.
Yeah, of course, because this is the we haven't done it because we needed the new protocol and new ethics, and this is going to be done this month. This is already accepted, and this is within the plan. So we have only done one doses, and you saw these doses reduced 90%. But the question will be, yeah, what happened with this 10%? It decreases or not? So this is something we could not do before, because of ethics. But this is extremely interesting and important, because if one of the motivation is that bladder cancer has the highest recurrency rate, right? So we need to observe this, check if this in animals is also happening. Yeah. So we are gonna do these multiple doses, multiple days, and imaging, of course. Yeah, this is very, very good question. Thanks. Yeah.
Thank you.
So we have another question.
Yes, okay, I'm Ilaria. Thank you for your talk, very interesting. So I have a question about, your vision of your business plan is basically to go to the clinical trials in about 3 years, right? So you are very close to the clinical trials if everything goes well. And my question is, what is the cost of the product? So when the product goes on the market, and do you have a vision of the competitors? So are there other companies that are basically doing the same thing, and you can compete cost-wise, or you are competing technology-wise?
Yeah, sure, absolutely. Let me share back the screen because that's see how I got there.
Probably 3 years to the clinical trial seems very optimistic to me, but I don't know.
Yeah, it always depends on the money, right? The money you
exactly.
got.
Since you are raising Series A, I guess the next step will be Series B for scalability.
Yeah, so these are all our plan, right? So we are planning to move, and this will be for the next 2-3 years. So develop this platform, right, to get to GMP-like, working on this, right? All the talks and regulatory for rodents. This we need to get there, right? We need to check a different type of efficacy in different type of products, and then we need to move the company from the pre-Series A to the Series A one. So this should take a couple of years. So competitors is more about the system, but here are the competitors. Yes, there are like actually the competitive landscape a year ago was almost empty. And now there's a lot of migration from prostate cancer to different type of cancer and using different drugs which are in the market for bladder cancer, right? Because they see that there's a huge market if there's so much recurrency, right? So, so you see my screen, by the way? Yes.
Yes.
Yeah. So Johnson & Johnson, then this Ferring, this was recently accepted, and you have Engine. There's a lot of competitors here, much more than what I am putting here, but indeed, some of the products. And there we know about a few clinical trials with different drugs which are jammed there. So this is competitive landscape is increasing, and you see these are important names that you can see here, right? Then the first clinical trial in between 3-5 years. I think 3 is optimistic, 5 it's realistic if everything goes well, and it cannot be much more because these clinical trials that we know and these companies are really advanced, right, from us. Then the next point will be the cost. The cost of goods is very large. I cannot tell you the numbers, right? Then the point is like whether you position as a first or second in the pipeline, right? And also if your efficiency is much higher. Some of the drugs which are done now intravenously or not systemically, let's say, they show like huge side effects. So then there's 35% of retinal disorders, and if we manage to, even if our product is more expensive, but we increase the efficiency, we have some hopes and numbers that we are doing these days as well, right? To show that it makes it very good product to jump into that. People are moving also to intravesical, which is where we are now, and that's also a very good strategy because we are following the same protocols in clinic. So it's a balance overall, as you see here, the platform adaptability, accumulation into more efficiency, and so on, right? We are more versatile than other products, but cost is a big challenge as well. Yes, it.
What is basically the quantity that you are estimating that a single patient needs to have like appropriate treatment, if you can share?
This number that you're asking actually is done somewhere here. Actually, we're going to make 3D bladder models in order to get this number. This is a very, very good question that needs a whole project, right? This will plan for 6 to 8 months to know how much we need. And that will be linked, as you see here, with the CMC development. Right.
In the papers that you basically shared before, low dose, high dose for the animal trial. So basically from there, you can extract some kind of estimation of quantity.
Oh yeah, there are two things, right? The fact that, I mean, you have a small blood here. You have the small blood from the mice, and then this will be the blood from humans, right? In terms of volume, you can make up these numbers. Yes, I agree. But in this particular step, we want to see whether this is a reality, right? That we can. So how much we need in order to fill all the volume of a large bladder from humans. So this is one of, I mean, you are very smartly asking the key question, right, which is in our research plan, like whether not only the particles, because let's say maybe you have a lot of particles, but still they don't reach the whole volume of a human, and they also can sediment. So we need to play a little bit with the fabrication, propulsion mechanism, and everything. So it's not straightforward. It's not just how you call it.
General technology fails for this reason, cost of the product, at least based on what I know. So I mean, sometimes investors, they ask about cost of the product and, you know, scalability as well, you know, how you're going to introduce your product in the supply chain in the future. Yeah,
these all the things that they ask, and we have these answers, and we are giving these answers to them. So this is clear. This is clear that needs to be addressed, right? And there are some things that I cannot disclose here, but well, we do have these numbers. Of course, you need to have a market analysis, competitive landscape, scalability, regulatory, and all these things, right? We have talked with 2 providers who can make the GMP and the GMP live batch. We know how much this will cost for our clinical. This is all clearly, right? Market research, as I say here, business plan, cost of goods, manufacturing revenues, all these things that I put here is what we are working, but of course, cannot tell you exactly numbers disclose here. Yeah, but these are really good questions, of course.
Thank you, Sam.
Yeah.
Okay, thanks. There is another question, please.
Hi, I have a couple of questions too. My first one is related to the enzyme. So do you produce your enzyme or is it commercially available?
Well, this is very smart audience we have today. Actually, we're having a very timely discussion. We do have two points here. First is we are producing our own enzyme, and we are testing commercial enzymes. Both things. We do have, actually, the postdoc that you saw, Daniel Sanchez, is working actively in my group, testing commercial enzymes and producing them. Okay, both. And we have this external advisor who has a whole company on the production of GMP enzymes, and we are gonna work with them on the scalability and everything. So with that, we need to decide whether we have a co-development with a company who has this, whether we modify the production of the enzymes to have the freedom to operate ourselves, or we also have a study on the patent like time, because maybe from the time that we get to the market, not the clinical trial, the market, then the enzyme, as we know, right, we can use it even if it's not ours that is commercial right now. So we are in these 3 strategies working. As you see, there are many things in parallel that we're working on very clearly. But the idea will be to produce our own enzymes, and we have checked, and actually we had a meeting today where we have seen that the enzyme we produce will bind to the enzyme to the particle, and they can still propel. So yeah, this is very important.
And my second question is related to the in vivo application, because I was thinking that the enzyme is exposed to the proteases from bloodstream. So is it degraded? And then how is it metabolized or excreted once the treatment is finished? What happens?
So the first point is that it doesn't go into the bloodstream, so it stays in the bladder. We have in our paper, we have I think it was like tens of animals in the supporting info where we check that there is no migration, there's no crossing of the ureter, and nothing goes through the bladder, okay, on the outside. So this is not a problem. Then second, whatever is not accumulated in the tumor will go outside with the urine. So that's why, as a proof of concept, the bladder cancer, I think, is ideal in an ideal system. And the third point is, to our surprise, we have seen nanobots moving over hours in vivo. And the current treatment of chemotherapy or immunotherapy takes 2 hours exactly, because the time that our bladder takes to be filled with urine. So no longer that makes no sense. So actually, in 1 hour, we have seen the whole efficiency on the tumor accumulation. So over the time, going to your question, over the time that we have the experiment, enzymes keep active because the nanobots keep moving.
Great, thank you.
Welcome.
So I don't know if there is any question here in the room. No? Okay, so please, I just want to.
Hello. Hello, Samuel.
Hello.
Thanks for your nice presentation. And today I came to know a lot of new things.
Yeah.
Dr. Arunangshu, joined from India. I working as a material scientist from a reputed pharma company in India. So from a chemist point of view, I want to learn or ask one simple question. So if we move for the plasmonic nanoparticle or polymeric nanoparticle or any iron-coated nanoparticle, among all of them, which one should be good for nano robot for use in the cell?
Yeah. So we do have, I mean, you can see in our publication is Nano last year, we had also iron oxide core, like core shaped particles, and they can be very good for guidance, right? You can also make bubbles with that. And you can also, maybe if you have magnetic field, you can use for heating effect, right? This one. Plasmonic might be good for imaging, right?
Okay.
If you want to use photoacoustic, if you want to use light as well to have thermophoresis guidance, or for actually, I think there's I put it here in my slides. There's a company in Israel, it's called NanoBotix, I think, or nanorobotics, where they claim that using plasmonic particles with light, they can also drill into. I haven't seen any result. I have just found the company, but is following your question, I think, for Professor Turner, and they induce heat locally in the tumor. So yeah, this material combination based on your background, I think is very interesting, and these are some things that we are doing. We are combining with light, magnetic fields, but also in the company. So this one thing is the lab, right? You can play along with that, but in the company, we want to keep it as simple as we can, because all the discussion we have, right? Then you go to a regulatory and this is a mess, right? People, and then what about the cost?
Because the cost-effective method, and also there should be some cytotoxicity factor during the cell that we should think about it, which type of the nanoparticle we can use it.
And there's also controversy between the gold. I mean, plasmonic gold, people like it, some people don't like it. There are some discoveries on results and clinical trials that are accepting. Some people at the fundamental level, they say try to avoid gold. So it depends on that, right? Same for probably iron oxide, whether it degrades or doesn't degrade. But I think the bladder is a nice place because it's confined, so you don't need to deal with many other things, right? That are, if your particle doesn't go through, right? If by some reason you drill into the lumen, then you do have a problem, right? But otherwise, the risk is reduced, right? In the bladder tumor, let's say. So that's interesting.
And my last question, so it is from curiosity I'm asking, what's the demand for a synthetic material scientist to work in your company? So one who know about the synthesis of the various kind of the nanoparticles, starting from polymeric or any kind of the coating or any shape modification using laser light and also photochemical etching. So this kind of the background, is there any opportunity to work with your group in future?
Yeah, absolutely. So we are opening 2 postdoc positions at least. Now we did open one last week where we are looking for people expert on synthesis of nanoparticle, enzyme functionalization, drug delivery system. This is one. We need another one who will be more on the physical chemistry on the chemistry of physics as well, on the material sciences, as you are saying, right, to develop to have a clear protocol. Once you go to the CMC, to the production company, right, you need to have a very clear protocol, and you need someone expert on these materials to iterate very actively with them, because otherwise the cost will increase dramatically, right, if you don't have the expertise in your lab. So yes, definitely we are of course having the background on biochemistry for the enzymes, drug discovery, but or nanoparticles, nanomedicine, material science, yes, indeed. So if any of you is interested, we are
Samuel,
very
I have the background from physical chemistry and did PhD from material science, and also working in nowadays from electroorganic synthesis for few molecules.
perfect,
So
yes.
definitely I share my CV with you. Let's see. Oh, thank you.
Yeah. Great.
Okay, so-
Very straight. Yeah, great. Anyone is interested as well? We are looking for motivated people in our lab, in the company, so feel free to reach me anytime. Okay.
Samuel, I just would like to conclude, and I have a question for you, please. It's a very fast question. So you mentioned that these nanobots are based on the engine and the chassis. So my question is, how easy is to drive the nanobots? I guess that when we are working with nanoparticles, the motion of the nanoparticles are based on the Brownian motion. In your case, the movement just is based on the power of the enzyme. But I guess that the movement in your case is based on the concentration of the fuel. So in this case, the concentration of the urea. So what's going on? Usually the nanobot is going to the proper direction. So I mean, the nanobot is going to the regions where the urea concentration is higher, or I guess that if in this region is the tumor is located in that region, I guess the nanobot is going to the proper direction. But maybe it could happen that the nanobot is going in the wrong direction, because in that area the concentration of urea is less than in others. So I don't know how easy it is to handle with this issue with this technology.
Okay, and you say quick, but I was not that quick, so I'll try to answer as fast as I can. So this is what you're talking about, is chemotaxis. So chemotaxis will be a dream in our field, and I tried this since 2013, and it's not easy to replicate, right? So okay, so we will. I can tell you right now that I'm not sure if there's any chemotaxis based on that. So the particles are moving because of this chemical gradient. Chemical gradient around the particle, chemical reaction. But then, as you saw now, we're moving for collective. This collective motion makes some fluid flow, this convecting flow around the particle, and then one with each other start to interact. And this is very stochastic, there's not too much control on that. I cannot predict the direction. So what is happening here is that the nanobots keep moving whenever they reach the tumor site from the bladder cancer, because they change again, they get trapped into the extracellular matrix, which is softer than the urothelium because it's filled plaque. Okay, very rigid, very compact. And then because of the enzyme reaction, as I said, like Helicobacter pylori, they change the viscosity, the rheology of the media, and then liquefy and can penetrate. Okay, where there is a chemical gradient or not, I cannot tell you, because this is a whole bladder, right? There's urea everywhere. So we don't need to control the direction in this way, in this particular case, right? 95% of the nanobots that we identify with track are in the tumor site, 5% or less are in some other places in the healthy tissue. So we don't need directionality here. But if we want, as the listener was here asking, we can use magnetic field to guide them if we want, but we are not doing this. Okay, there's no need for that. But the chemotaxis is a whole topic in our community that no one has proved it in vivo, I guess. It's more in vitro experiment with microfluidic chemical gradients. We have seen this, but in vivo is difficult to replicate. Yeah.
Okay. So thank you very much. So it was very interesting lecture. Also, it was very interesting questions for the audience. So I guess as chairman, I'm glad to share with all of you this afternoon. So please, Samuel, continue with this research. We'll be happy just to follow your research maybe in a year or so, just to see what's going on with the nanobots.
Yeah.
So thank you very much, and all the best in the future.
Thank you for inviting me and for the very interesting question. It was a nice discussion. Yeah, thank you.
Okay.
Have a nice evening. Bye.